Hi, everyone. We've got a tight schedule today, so we're going to start right on time. My name is Steadman. I'm a bioinformatics scientist here at Elucidata. I'm joined by Manimala, who's one of our on our product team, and we today are going to talk about querying heterogeneous biomolecular data on Poly. We're going to do some pretty cool searches and really dig into what about the way we've structured data on Poly makes it different from doing these types of searches at the source, and the benefits that come with that. Manimala, do you want to introduce yourself at all?
Hey, hey folks, welcome to Poly Demo Days. I'm Manimala. I'm a senior product manager at Elucidata, and let's have a very good session. Feel free to ask questions anytime you want. Yeah.
Great. Okay. So if you guys have logged into a Poly Demo Day in the past or talked to us at all, this probably will look familiar, but it bears repeating because it's going to set the tone for what we're going to show. And really, the reason that we at Elucidata want to talk so much about querying and data structure is because most biomedical data that's out there is unfair. FAIR, again, is an acronym that stands for findable, accessible, interoperable, and reusable. And the cost, there's a big cost associated with our biomedical data being either proprietary or public, not being fair. They create a lot of bottlenecks. And so what does that really look like? The data at the source is scattered across different places. Sometimes those repositories have limited access or restricted access, which can cause problems. And it's really hard when you have data in many different places to keep up with new and emerging research. Once you're able to get access to the data, the problems don't start there. The data is often unusable. And what I mean by unusable is that it's unstructured, it lacks a standardized metadata schema. The data processing pipelines aren't consistent, and sometimes you lack information describing those pipelines.
Hey, Maya, one sec. We
yeah,
can't see your slides.
You can't see my slides at all?
No.
Oh, great. Okay, 1 second. Let's try this. Thanks for letting me know.
Oh, of course.
I would have gone through the whole presentation. Okay, you can see my slides now?
Yes, they're up. Yeah, they're up.
Okay. I apologize, my bad. I won't repeat myself, but what we're talking about is the problems of the data at source and just some cute infographics to go alongside. It was great that you told me that because I was about to actually show examples of the data at source. Okay, jumping back in. We're on what makes the data unusable. As I said, it's unstructured, it's lacking certain metadata. The data processing pipelines can be very inconsistent, and sometimes you don't even have the information about how that data was processed. And it's not easy to store it, right? So when we're talking about public data repositories, we are talking about large amounts of data. So not just 1 or 2 data sets or even dozens of data sets, but millions and millions of data sets. And it becomes very non-trivial to store and organize and be able to find that much data. And then finally, there's just a lack of, as I said, data engineering, structure and tools to really make the most of the data that is available. And when you do have the tools, you often need some pretty intense expertise, some domain expertise and domain knowledge to make the most and understand those data structures and actually be able to take the data out and use it. So what I'd like to start with today is just kind of reviewing what the data would look like at the source. And we'll start with the data schema, right? So there's kind of 2 main problems that we covered: the way the data is structured and then how it's annotated. And we're going to cover both of these with a few examples. So let's tackle the data schemas being inconsistent at public sources first. What we have here is just some snippets. We've taken some screenshots from GEO itself On the top, we've got a particular GEO study. And what we found when we went to this page is that the series matrix, so the actual data, wasn't even in one table. It's split across 3 different tables here. So right away, I don't even have one. I'm not even talking about dealing with one file. I have 3 different files that I'm going to have to put together to use. Another common scenario that you would see is if you had 2 different data sets, they may not have the same file format. And we'll talk a little bit about the problems that that causes in a second. And then on the bottom, what I have is we're looking at we've opened up the data matrix and we're looking at what's inside that data matrix. And our row level data, the gene identifiers, they're not consistent across 3 different data sets. So if I wanted to compare, study 1 to study 2, I wouldn't get very far because the gene identifier isn't the same. So I would have to first, take my entree ID, my gene ID, and my ensemble ID, and find do some sort of lookup to make sure that I can match them, and then create a key so that I know that I'm comparing the same thing across these data sets. And so what we found is that scientists spend hours to days taking these data tables, merging them together to make them ready for downstream analysis, and that of the data that's on GEO, only about 3% of it is machine readable from the get-go. So that's a big problem. And then just to kind of drive home this point, the problems don't start at just the way that the data tables are laid out. The metadata itself, so GEO stores the metadata in a series matrix file. So it's separate from the data itself. So that is also a problem, that your data and your metadata are in 2 different places. So far we've got multiple data files, separated metadata from our original data source. And now when I'm looking at the metadata, this is just a quick example, but 3 different studies here, and each of the sample IDs are actually labeled with I don't even want to call it an ontology. They're labeled with something that was specific to that lab, right? The source wrote the publisher wrote what made sense for them. But for me as an outsider, I would have to kind of translate what that means. And I can do it as a human, but a computer definitely couldn't. So if I wanted to process all of these data sets together in the same pipeline, I've got 3 different ways, all just to indicate which replicate it is. And that lack of controlled vocabulary doesn't stop at the sample name. It continues through every column of that metadata, where you're going to see, a row like this one's got spaces in it and this has got underscores. And so is this hyphen going to mess up my pipeline, right? These are all tiny things, it may seem, that you'd be able to handle, again, on that 1 to a dozen data set level. But when we're talking about data at scale, when we're talking about large volumes of data, it makes it unusable. And so we've got an example of a simple search here that just trying to find all of the data sets that met these requirements that would answer this question could anywhere from days to weeks to months. It just becomes completely unmanageable. And so this is the challenge, right? This is kind of the summarize. We've taken all these steps. If I wanted to do any analysis, it's just an example of a timeline of what you'd have to do to prepare any one analysis or any one data set for analysis. And what we found is that researchers are actually spending their time not doing the research, but cleaning up the data. And that's what we've set out to solve here. And Manimala, I'll invite you to comment on this, because I know you've had a lot of experience also in this realm of cleaning the data.
Yeah, so it's really a pain, because you will have to first spend a lot of time to clean the data, understand the data structure at source, then spend a lot of time to clean the data to make it usable for any kind of application. It could be a simple dashboarding or a linear regression or a statistical analysis that you want to do. All these downstream analysis have that power, or that they generate the insight that you're looking for, only if you are using very clean data that is harmonized. And hence people spend a lot of time trying to do that. And at Elucidata, we understand this pain point very well, and hence it's our goal to fulfill this unmet need for the industry. Yeah.
Yeah. So with that, I know, again, if you've logged in before, you've probably seen this exact slide, but it's just so important. It really does bear worth repeating. Let's talk about our technology and what we're doing at Elucidata to take this, the mess of the data that can be at source, and what we've done to make it ML ready. And so every data set that's on Poly has undergone 2 key steps to take it from unfair to fair, or ML ready. And that first one is our data engineering. Like we said, we take all of the different file forms that you could expect to find from the data at the source, and we put it into one standardized tabular data schema. And we're going to spend a lot of time talking about that data schema today. So I'll just leave it at that for now and focus on step 2 for a minute. And that's the metadata harmonization, right? And so this really speaks to the findability of the data that's relevant to you. What we've done is we've gone across all of the data sets. We have chosen a set of standardized ontologies that we're going to use on every single one of them, right? And so whether you're looking at a data set from TCGA or GEO, you're going to have the same disease terms, you're going to have the same drug terms, you're going to have the same cell line terms. And what that's done is it enables users to not spend time doing that find replace, doing those keyword matches, and you have confidence that all of the data that you're there, when you put in a search term, you're getting every hit, and you know you're getting every hit because we've taken we've done that legwork, we've done that cleaning, and we've put in those proper tags. Now, what about missing data, right? As I said before, or maybe I didn't say it, but you guys probably know, a lot of the ontologies or the annotations can be completely missing. Sometimes it's not there at all, right? Like it maybe says in the abstract what the paper is, but at the sample level or even on the data set itself, it doesn't have the tags that really describe the sample. It's incomplete metadata. And so what we've actually done is gone through with those standardized ontologies and imputed and filled in the gaps of that missing data. So not only is it the same, no matter where it comes from on Poly, but if it was missing, it's now been filled in. And what you end up with is a really complete data table. It's very intentional and very easy to read. And now you can do these very powerful searches that often you'd have to kind of check and recheck and use a different syntax very quickly. Okay, so let's focus a little bit deeper. Let's dig a little bit more into the metadata harmonization. And Manimala, let's talk about kind of what the process looks like. And we'll start with what's done outside of Poly.
Yeah, thanks, Maya. So I'll spend a couple of minutes trying to explain what is done outside Poly, what is the industry convention, and then what we are trying to do at Elucidata. Outside Poly, people typically use either manual curators, or they would use something like a semi-supervision model. Now, there are 2 caveats to this. If you are using a manual curation method, your process is not scalable, because with every incoming data, you would need manual curation for that many fields that you want to curate, and there is a cost involved to it. The second thing that you can use is a semi-supervised model, and these rely heavily on hand labeled data, which is essentially labeled data to train the models. And these training data they require a lot of training data. To generate those lot of training data, you would again have to go through that manual curation process so that the training data is accurate. Now, all this involves cost. So within Poly or in Elucidata, what we have done is we've taken a very different approach, which is a combination of weak supervision and active learning. And what we are trying to do here is we use less number of data sets, but high quality data sets to really build our models. And once the models are built, these models are only scalable and they get better with time as we see more or as we are challenged with more data. If I have to leave you guys with some numbers, then I guess a fair estimate that it reduces the manual curation effort by up to 70% would be an estimate that we can achieve compared to things that are done outside Poly in terms of curating data sets, and harmonizing it and making it usable. Maya.
Yeah, thanks. So we're gonna really utilize the metadata harmonization in our demo, and I know I'm building it up, and I think you guys are really gonna like it. So it's good to have this background and understand the work that's gone into it and how we're getting these tags. Okay, let's shift focus and talk right before the last thing we need to cover before we go to the demo is actually talk about the data structure. And so we've curated the data in a number of different levels. We'll refer to it a few times throughout the notebook demonstration that we're going to go through, but it's the data set level, the sample level, and the feature level. And so maybe before we show examples of what that looks like on Poly, again, if you could just give us a little bit more detail about one particular file. So what we're looking at here and how we use that to help us make our searching faster.
So what you see here is a GCT file, and let me first take you guys through the process of storing this data at scale, and that is something that powers our powerful searches or the queries that we will show in some time from now. You will have something which is a data set level, and it comes with its own metadata, which is the global metadata. Then each data set can have n number of samples, and each sample can have its own metadata, and each sample has its own data matrix, and it can have feature level metadata. So now imagine the scale at which you will have to beef up your infrastructure to enable the kind of querying that we will demonstrate in some time. Also, a primary requirement from any search or querying standpoint is that they should be compatible in the sense that I should be able to query across repos or across data repositories. So what we can enable at Poly, because we follow a standardized schema, the querying across repositories for a given disease, for a given data type, you name it, and it is seamless. You will not have to understand the data structure at source. We've done that work for you. All you need to understand is how one of these queries are run, and the queries are scalable. So that's how we've done it. And to just let you know, we host about 1.5 million data sets on Poly across 26 data types, and ranging from transcriptomics, single cell, mutation, copy number, you name it, and we have all the important data types on Poly. Over to you, Maya.
Yeah, all right. With that, let's go ahead and get to the notebook. So just guys, give me 1 second. I'm going to stop sharing, and then this time I promise I will remember to actually share the slides again, the screen again. Oh. Sorry. We need to like hold music while I'm pulling it up here. Notebooks visible?
Yep.
Great. Okay. At least I didn't get halfway through before I started it. Okay. So I am actually in our PolyPython notebook. We'll talk a little bit about the features of this notebook as we get a little bit further into the demonstration, but you can see it looks just like a Jupyter notebook that you would launch from your own local machine. The only difference is I've done it from inside a Poly Workspace. If you don't know about Poly Workspaces, it's a space provided on our platform that you can store and organize your data and your analyses. They're all saved in one place, can launch and relaunch. It's very collaborative. Manimala and I have been working on this notebook together over some time, and you can change the permissions of anyone who has been added to your workspace as you go. So it's a really nice feature. Saves all your information. You'll see the autosave things popping up every now and then. And it's really great. And PolyPython also has a bunch of packages that are needed for downstream analysis already installed, so the environment's ready to go. But without any further ado, I'm going to scroll past a lot of the setup for you guys. We've done quite a bit of setup. We've imported the necessary libraries here so that and define some functions so that the searches that we're going to do are ready and go a little smoother. And there's some great documentation, so it's really easy to set these up on your own and define your own as you're learning to use PolyPython. But let's start really basic, but still pretty cool. As Manimala said in that last slide, because all of the repositories are stored into a single standardized data schema, we've been able to very easily search across multiple repositories with not a lot of work. And so what you're looking at here is I'm looking for mutation data sets in breast cancer across 2 repositories. So we're going to look at CPTAC and TCGA. And if you looked at the query here, you'll see in this first one, I'm going from CPTAC data sets, and I'm looking for the dataset ID, description, and disease, and it's got to be breast cancer and its mutation data type. And almost the exact same syntax with just maybe a few changes are used for searching for TCGA data sets. So right away, really easy, can look for both at the same time. And what I found here is I actually got 863 data sets for this result. And one,
so,
yeah, if you want to jump in.
Yeah, so one thing to take a note is that what it is showing is the amount of data it has scanned in the time that it has scanned. So your cost of querying, if someone is worried about the cost of querying, is really low. And the second thing is, in case so if you can see in the first example, and we've chosen this example wisely to demonstrate that even if in a certain repo there are no data sets matching your constraints or the arguments that you're passing, it will not throw you an error. You will still be able to see data sets from the next repo, in this case TCGA. So in the first example that we're showing, you're seeing all the data sets coming from TCGA because CPTAC does not have breast carcinoma cell lines samples. Over to you, Maya.
Yeah, no, that's a great thing to point out here. Okay, so for our next search, so pretty basic search, but still very effective, very fast, and nice to be able to search across 2 repositories. So we're going to do the same thing, but 2 different repositories here. So sticking with TCGA and looking for GEO, and now we're going to look for transcriptomics data sets. Again, changing repositories, syntax, very similar. And very quickly. So just a very low cost search, and we're actually getting over 3,000 matched results here.
And in this case, you are getting results from both GEO, which you can see the GAC IDs, and you are getting results from TCGA. So it is essentially taking a union of these 2 repos for the data type mutation in this. So you've expanded your search in some terms. Yeah.
Okay. I'm not going to review this again because I've said it like 3 times already, but I am going to mention that you should pay attention to the data schema at this point, because we're going to go a little bit deeper. So we've been doing that data set level search, and now we're going to jump down, and I'm going to focus on GEO, and we're going to go to the next level and do a sample level search. before we do that, one really nice feature of PolyPython is if you ever don't know how you want to search, so this is or what is the proper syntax that you should use for searching, we've got this great describe feature, and it works for all of our repositories. And so in each 3 levels of the data schema. And so I can just ask PolyPython to describe my GEO datasets, and what I get is just a list of the column name and what the data, how the data type is stored in there. And that's going to inform what syntax I need to use for my SQL search. So just a really handy feature to keep in mind as you're writing your queries for your own research. So as promised, let's do our sample level search. So to set the scene, oftentimes it's not enough to just have a disease of interest, right? There's usually some more criteria that may not be indicated at the sample or at the data set level that is important for the research that I'm doing. In this scenario, I want to only find data sets that have both breast tissue, so breast tumor, breast neoplasm. In this case, we're using the mesh ID of breast neoplasm for breast tumor, and normal data sets. And that's because I want to look at the difference between my normal tissue and my diseased tissue. And that's sort of the sample level, right? Each sample is tagged with a disease. And one of the really cool things that we've done on the Poly platform is intentionally tag those normal samples. So it's not just disease and then a blank, or disease and control, or some other term. We've gone through, and anytime that a sample is a normal tissue sample, it has been tagged as such. So it makes it really easy to look at data set or look at the sample level, find those results, and narrow your the data sets that would be relevant to you down. And that's all we've done here. Is I'm saying, okay, return my data set ID, look in my sample level, and I need it to contain breast neoplasm or normal. And I'm just saying here that it has to have breast neoplasm, right? Because there'll be normal data sets and GEO that aren't breast, but I need it to have breast. So I'm setting that filter of one of the samples in the data set has to be breast. And I do this search, and again, very quickly, I get my results, and now we're reporting the actual rows of samples. So if you look at this table here, what you'll see is the data set ID and the actual sample, the sample name tagged with the GEO sample name, not some random syntax that is specific to the lab that generated that data set. We're using the ID from GEO, and you'll see the disease tag in this column. And so this is great, but I still have 37,000 rows, which is a little bit clumsy. And so I want a more concise table. And so all I'm asking to do now is to aggregate that data into a table that's a little bit more readable. And so I've aggregated it by the GEO dataset ID here, and then counted how many of the samples within that data set are breast versus normal. And so if I was trying to, I wanted, depending on what my goals are for my downstream analysis, I may, need to have data sets that have a high number of samples, right? So here's 112 versus 30, and that might be the power that I need, or maybe I'm looking for something else that's more specific. But as you can see, you can go through and very quickly get these concise lists to find your data faster, as opposed to needing to go do your search and then read all of the abstracts. Because these tags have been implemented, we're just reducing that time to finding the relevant data. And I'm just going to keep running, but if you have things you want to add in, Manimala, please feel free to jump in. Otherwise, I'll just
let's go ahead.
Keep running. I know we're a little quick. We have a lot to show and not a lot of time, so I'll skip over this. This is, I just printed out the data matrices just to emphasize again that it's very organized. This is all one file format. Anytime you pull out one specific data set, I've got 90519 here. Instead of it being in multiple data files, I have all of my metadata in one neat table in a tabular format, very easy to search. And the same with the actual data series or the data matrix. All the same gene ID, no matter what gene data set that I'm using. And it's always tagged by the GEO sample ID at the top, not not the name that it was given at source. It's given the controlled name. Okay. Now it's time for a little bit of fun. So we're still staying within GEO now. We're going to move out of it in just a second. But the one last thing I want to show you in GEO is we're going to move down to that feature level. So this is something that's really specific to Poly. It's really, really challenging to do these things without the structured data schema that we've set up and without the beautiful metadata harmonization. These two things together allow me to look at individual rows within a specific data set across multiple data sets. And that's what we're going to do. So in this scenario, I've got a specific gene that I am interested in. For whatever reason, some in-house research has told me that NRP1 is really important for breast cancer. And I really want to see, I found my data sets that have normal versus disease, and I want to see the expression across multiple data sets of this gene. And that's what I'm going to do here. So I'm going to go in, I'm going to pull out my data sets, and I'm going to look at the sample level. I'm going to subset my samples into two different categories. So I'm going to say, okay, if it's normal, look in the disease term, find normal, put that in one group. And if it's diseased, it's going to be breast neoplasm, put that in my disease group. So I'm taking my samples and splitting them into two different bins. And now I'm going to look at that feature level, that row level, and I'm going to pull out a specific gene. And then I'm going to plot it, right? And I do that here. I'm looking at these two specific, my extract gene and my gene is NRP1, and here are my two different data sets. And you can see the times here, how quickly we're going through to plot this data. And right away, with not a lot of work, I can know whether or not a NRP1 is differentially expressed in my breast tissue versus my normal tissue from these data sets. And I haven't even left my notebook, my Poly notebook environment. It's very quick. I would make the conclusion from looking at these plots that it is not significant and I would move on, but sometimes the no-go decision is as important as the go decision. So I know I wouldn't need to take these data sets any further into my downstream analysis. They're not worth my time and I'm going to move on. Okay, that was a lot. Keeping an eye on the clock, we're going to move right along, keep rolling here, and we're going to talk about mutation data. So if you remember in the beginning, we did two different data set level queries. We looked for mutation data in CPTAC and TCGA. We didn't find any in CPTAC, but we did find a good amount for our breast cancer in TCGA. And we're going to stay at that sampling feature level and start querying that data now. Again, I'm using this describe function, but this time I'm at TCGA in the feature level. So I can just get a quick idea of, the column names that I have to search by and what the data within those columns are. So now I'm well informed for how to set up my queries.
So just to add,
Yeah.
Maya is talking about describe again and again. So when in doubt, describe is your best friend on PolyPython. Just go ahead, use the describe function, see what the schema looks like. And then it just makes your querying downstream very helpful, very seamless.
Yeah, it is. You don't have to memorize any of it,
yeah.
Which is really nice because I would never remember any of it.
Yeah.
Okay, so in this scenario, you'll see this again. I'm going to harp on this a lot. The syntax is very similar to my data set level. I'm using the same type of form across everything. So the reason I keep pointing this out is we're trying to highlight that I don't need domain knowledge from source to source to source, from level to level to level. We're using a very similar form of search, no matter how granular or how broad we're looking. And that makes it a much lower barrier of entry to access the data and find the data that we need, right? You're not relying on that one person in your lab that is like the whiz at TCGA, right? Like anybody can come in here, and if you can learn SQL, you can find this data. And that's really powerful. So what we're doing here, looking for mutation data sets in TCGA for one disease, again, breast cancer. And I want only tumor samples.
So
Did I miss
yeah,
anything? Yeah.
it is important because TCGA can have both normal and tumor samples, and in this case, we are running an example so that we can fetch feature level or variants in our diseases of interest. In this case, the disease of interest is breast cancer.
Yep. Yeah. And so we've done this search. We pulled it out, and again, here we are with our 863 rows, but these are all in TCGA. Each data set is a sample, and so these are all in different files, and I want to look them all together at once so I can find the ones that are most relevant to me. So we're going to go ahead and merge them into a single data frame, and that's what we're doing here. And again, now I'm going to pull that out and print it. This is the same search as you've seen above. We are just redefining the terms here. So I found my data sets that have, breast is the tissue, breast neoplasms mutation, and I'm going to go ahead and download all of those data sets. Do you want to talk a little bit about why we're able to do this so quickly? Because it is a lot of data sets.
Oh yeah, that's because that's because it's the power of polycompute and the parallel processing that we can enable. It is really fast, and the merge does not take time at all. So yeah, that's the reason. Yeah.
Yeah. And then so we've merged all those files. So all 863 are now in this one table. And that's what we're looking at here. We've got our all of the metadata from each one of these samples, your patient barcode. I don't know what else is really interesting.
You'll have information like age, you'll have information like ethnicity, you will have the primary sites, you will have the tumor stages, you can and the sample type, any information that is required to understand the patient history from where this particular sample in TCGA is derived. You can all look at this in a single data frame.
Yeah, this is actually the stage, the cancer stage is a question that we get a lot. And if you were to do this on TCGA, you'd have to look at each file individually, right? These would be all in different files versus here, because we were able to merge it into one data frame within the Poly environment. I now can take this data table and search and filter specifically to the stages that interest me to get those IDs, right?
Yeah.
So that what we've done here, we've created our own little data matrix. And so say I've gone through and I've found a data set that is of interest. I have it highlighted here, right? This is the natural, we're just speeding up the natural progression of a research analysis that you would do. I've looked through my data sets. I found that I'm really interested in this particular data set on TCGA, and I want to pull that out and I want to look at it specifically. So here it is. Here we're looking at the feature level, and this is what you were talking about. How about on TCGA, it's very unique because there's so much metadata on that feature level, right?
Yeah. So essentially what you see in this table is something called as a map table on TCGA, and it gives you the information on the variant per se. So in this case, you can see in which gene this mutation is, what is the start position, what is the breadth, which is the variant effect predictor. You can see in which center this sample was derived from. You can also check for any sequencing related things like what was the sequencing depth of the alternate allele, what was the sequencing depth of the normal allele. You can go to any level of granularity from this one table for every sample. And this you can now, if you've selected n number of samples, you can again use our merge functions to merge all those sample features in one data frame to look at it in one shot, which is not done here, but you can do that.
Nice. Okay, So now I'm going to go ahead and I'm going to pull out this data frame or this particular data set, and we're going to look at it a little bit more in depth and do some plotting with it, I believe. And so that's all we're doing here is we're calling this repository, we're calling TCGA, we're pulling out this particular data set, and I'm pulling out the GCT file. So I'm downloading it and loading it into Poly. And so you'll see here, this is another really kind of fun feature of the PolyPython or the Polyglot notebooks is all of the code that you've seen so far has been in Python, but this notebook could actually handle 4 different languages. And so, for the plotting that we wanted to do, we wanted to use math tools, and that's in R, so it's an R package. And right within the same notebook, we can just switch our language here. And so I've indicated now, and you'll see I got a different color. And we're going to continue in the same space, but switch languages here. So we're in R, and we're just setting up our plots. Again, we pulled out our data set, we downloaded it and loaded it back in. And here we are. And so what we're looking at here is an oncoplot and a TITV plot. And right on the top is our just the summary of the data that we're looking at for this particular data set. And I'll let you walk us through the features of this plot. I can zoom in for you.
Yeah. So so the first plot that you see, it talks about that most of the mutations or the SNVs are not missense mutations. There's one nonsense mutation and one splice site variant. Most of them are SNPs, which is the second plot, and it tells you the SNV class, which is whether it's a transition or a transversion. So T2G is only one, most of it is C2T. And on the plot below, what you can see is the variants per sample. In this case, in the summary itself, you see that there are 36 variants. You also do get to see the genes that are mutated in this particular data set or the sample from which this data set was derived. Yeah.
Nice. Okay.
This is a long plot, and we can skip this in the interest of time.
Yeah, we only got about 5 minutes left. So, okay. So the last example, I didn't mean to cut you off. Sorry, go ahead.
No, that's fine.
The last example we want to show is really cool. So we're going to search. We had a lot of fun making this. We're going to search across TCGA for a particular gene. In this case, TP53. We're really obsessed with this. We want to find it. And I want to find all of the data sets in which there's a mutation for TP53 in TCGA. And that's what
to give a context, TP53 is one of the most mutated genes in any cancer. So researchers probably want to look at in a given repo how many samples have TP53 mutations, and we've used TP53 here because, as I said, it's a tumor suppressor, highly mutated in every cancer, almost every cancer. You can select any gene of your liking, and the results would look the same. Depending on whether it's present or not.
Yeah. And again, I'm going to sound like a broken record, but I'm using the same syntax here. And now I'm looking in a completely different way, right? We've done a lot of top-down searching, so data set to sample to feature, but now we're kind of going the other way. I'm saying I've got this particular feature that I want, and I want you to return the data set ID and the information about where this mutation is, where I can find the data set that has this mutation in it. And that's what I've done. Or that's what we've done here. So we've gone through, we've looked through our T53, and we're left with a data table. And if I just open this up, what we find is all of the rows within TCGA. I've got my data set ID, the disease, and if I scroll down, you'll see it's not just breast neoplasm, it's just alphabetically what comes first, but I've got multiple different cancers that are listed here. I continue scrolling across. I see the protein position, the amino acid change, the impact, the variant class, consequence and then the name of the mutation here. And it just took over a minute, which is just a lot faster than you'd be able to do. Like trying to do this type of search, although it's possible outside of Poly and on TCGA, it's just not as simple, and it certainly isn't as translatable. I could replace TCGA with another repository and do a feature level search in this way and get a very similar table.
Yeah, so would
Yeah,
just like to add one point here. So this table is fully customizable, unlike any other GUI application, which probably many people use over TCGA, which are out there over TCGA CBio portal, where you really cannot customize the fields that you want to also add to the view. So what you can do is in the query, you can specify which additional fields you want in your data frame, and then run the query, then hit the query, and you should be able to see a modified data frame with all your new data frame. So yeah.
Yeah. Okay, so we are just about out of time here. You want to just quickly talk about the last thing that we did, and then we'll
yeah.
sign
So
off.
This is really simple, which people like doing, and I personally also like doing. So yeah, so there are a couple of mutations, actually 10 hotspot mutations in TP53. Detailed documentation or a paper is there in cell death and differentiation. So the idea was to look for one of that mutation, which is tyrosine at position 220 converted to cysteine in the protein in the TCGA dataset across any dataset. So I'm not restricting myself to breast cancer or any other cancer. I just want to see how many datasets have this mutation. It essentially becomes very helpful when people are trying to understand the frequency of mutations or the occurrence of such mutations in large number of data sets, especially in repositories like TP53. There's a very simple query that you can see, and it throws you a data frame that gives you the number and the dataset IDs, and of course, the cDNA chain that you're seeing here. Yeah.
Cool. Thanks. Okay. So I think we went a little bit over. I'm going to go ahead and stop sharing here. But if anyone has any questions, please reach out to us. We'd be happy to try to take up your questions. You can email me at maya.steadman@gmail.com, or not Gmail, at Elucidata. You can email me there, but I probably won't respond. At elucidata.io. And Manimala, go ahead.
Yeah, and thanks everyone for joining in and watching this video later when this is up. So this is Manimala signing off, and as a parting remark, do feel free to follow us on our GitHub page. You can see a lot of important documentation, things related to PolyPython, our programmatic access. Feel free to even reach out to us on that page, make pull requests, and we'll be happy to take those things. I'm sharing the link here, and my email address is manimala.sen@elucidata.io. And yeah.
Manimala, thanks so much for joining me today. I had a great time both building this and running through it. We've had a lot of fun.
Same here.
Yeah. And